different. So that is also people have done it. It is established. For example, the one we are telling, it is a Nature paper where they have shown, basing on the sexual behavior, they have characterized the short-term and long-term mutants, and the players involved in the short-term and long-term memory in Drosophila and human, they are the same guys. That is a beauty. For example, memory, the famous calcineurin, the great Timtuli from Cold Spring Harbor, his system is Drosophila. So I feel actually it depends on the dam, it depends on the ability of the researcher, any kind of model system, right? For example, C. elegans. C. elegans, it is, it just crawls, but still there are people who characterized multiple phenotypes, maybe some kind of crawling, they say it is a neurodegeneration, which I do not understand because that is not, so therefore there should be a way, depending upon that's what we feel. If there is any other question, we will take it. Yep.
Can we study cancer using Drosophila?
Which cancer? There are multiple Drosophila cancer models are there. For example, Sir was talking about NF-kappa B. NF-kappa B, you can check the literature. The contribution of the Drosophila for the NF-kappa B per se and the players, the friends and foes of NF-kappa B, I don't think any other model system other than Drosophila has contributed. See, what happens is your human story ends up the moment you identify the mutation. That is a very important point we need to understand. Yes, most of the time people who work with the clinical samples, that is a popular point. For example, you identified the alpha-synuclein in human populations with the pedigrees, DJ1. The moment, okay, you identify the mutation, after that what? How it is going to happen? Yes, in general, overall, your nutraceutical food story, it looks good, fine, no problem. But there are n number of the diseases because of the genetic mutations. How are you going to treat the genetic disorders? You need to understand the mechanism. What is mechanism? If a fellow is there, he is not working. And who is the guy stopping him? Whether he is upstream or downstream. If somebody is helping, who is the fellow helping him? For example, by overexpression of the heat shock proteins, Nancy Bonney has demonstrated protection of the dopaminergic neurons. So he is a friend. So it is important to identify the friends. Who are our friends? Same thing like real life. How are you going to identify the friends? Either through biochemistry, which may not be very accurate, at least we strongly feel if you do the HPLC and you are measuring it, if you run the 2 times, people who have done it, they know that 20%, 30% variation among the 2 runs from the same brain extract. So best way is suppressor and enhancer. I'm not saying only one should do with the Drosophila. No, that's what the scientific community has done. That's how novel genes in the cancer, novel genes in the neurodegeneration were figured it out. Best example is your period and timeless. So the point is, there is no other way. As a food we will say it, curcumin or some other thing. But if there are genetic disorders, only way to understand mechanistics is the model system. There is no, I'm sure Bharat sir would admit with that. Unless you understand the mechanistics, there is no way you can figure it out, drug targets or novel drug molecule screening. Unless you know that, like people, they know NF-kappa B, you can screen it. You can just feed, curcumin you feed it, anything, gerinol you feed it, some other plant extract you feed it, NRF2. Empty number of the screens were performed in order to understand the NRF2 regulation, because NRF2 also involved in multiple kinds of cancers, neurodegeneration. So only way is model system. Did I make my point? Yep. Yeah.
Can the human brain be rewired?
Rewired? It rewires every second minute. Yeah, it is an ongoing process, rewiring of a human brain, not only human brain, even Drosophila brain. You just take the guy, put into the dock and bring him into the light, or give a minute shock, and there is a way to scan the Drosophila brain. So rewiring is a continuous process. When you You know, rewiring is not a separate phenomenon. You have some experience, you see a great cloud or a mountain, rewiring is it's ongoing process. It is not a separate mechanism. Unless there is a rewiring, continuous rewiring, where is the perception of the experience? Yeah.
In Drosophila, can humans use movement protocol other than flying and walking up there as you saw yesterday in the tube? Other than these two things, any other motor components are there which you study or which you make to plan to study?
That's what. Maybe you missed the first part of it. There is also a way, we were talking about in 2017, two people got the Nobel Prize using a fly model to figure out the circadian rhythm. Circadian rhythm means how animal responds to the, maybe it may be annual rhythm or seasonal rhythm or day, night, light and dark cycles. So there are assays depending upon the question. Depending upon the question, you want to see the mobility defect one way, you want to see the circadian rhythm and the fly movement, and even the people, how Drosophila sleep pattern. There is another kind of rhythm, So that's what is, you are scoring the phenotype depending upon the question you are asking. So what is important is not your method. In the experimental biology, the power of experimental biology is the way you do the experiment. How do you do the experiment depending upon the question? So question should be clear. Answer, it's not important. Did I make point? So what exactly you wanted? For example, circadian rhythm, that's what I said. Mobility defects, I said it. Or Alzheimer's phenotype or some other phenotype. Depending upon that, it has to be, if it is already there, we will take advantage of it. Otherwise, you need to make a protocol, develop a protocol or characterize that particular phenotype. So depending upon the question, it needs to be worked on. Right, no? Yeah, any other?
Sir, you mentioned about
Sir, what you said is correct only. Actually, it's my mistake. We should have asked them before we go, no? The questions, before we left, we should have definitely asked and we would have gone.
Sir, you have mentioned in one of your slides the relationship between the estrogen hormone and the onset of
can you take it a little bit away from you? It is resounding. I cannot, it's like a horse tying with this one.
The relationship between the estrogen hormone and the Parkinson disease, onset of Parkinson disease.
Oh, okay.
So
That's
can you please elaborate on that?
No. See, the idea is like this. What in humans people know all the time, though we say everybody is equal, even for a disease everybody is not equal. Inequality is an inbuilt mechanism in not only anywhere and everywhere, even biological processes. The way the male brain responds, the way the female brain responds, it is different. It is important to understand. So the point is, the prevalence of the Parkinson's disease, like now demographically in certain populations it is. So that may give some clue. Similarly, male and female, in case of the female, it is less. Not only this, there are many diseases in female, it is less prevalent. There are some maybe, like there may be more. But the point is, then why? So people make, when we do not know, we always make intelligent guesses. What is the fundamental difference between male and female? Maybe the hormonal profile. They think that estrogen is neuroprotective. But in lab at least we discuss, we have a counter argument. Estrogen may not be the reason, as popularly people have been thinking, for the following reason. Because Parkinson's disease is late onset. By the time 55, already menopause is set in. So where is your estrogen is conferring the neuroprotection in humans? This is my feeling. But it is not that, that is the last slide. But it is not that when your dopaminergic neurodegeneration is there, you may be seeing the phenotype in a male or female when there are 50, 60, let us say, but there is a progression of the degeneration over a period of 15 to 20 years. Perhaps for the onset, maybe if estrogen is neuroprotective, we do not know how exactly it works. But you get the logic? When some disease is happening at the 55, 60, okay? Then what is that? In menopause already is happened, right? Then where is the estrogen neuroprotection, conferring neuroprotection? Having said that, how the development, in the development, there is also progression of the disease. It is not that all of a sudden a disease is happening, right? When 60, if it is showing up, maybe the process started when it is a 30, when it is 40, isn't it? But when 30, estrogen profiles are different. So there is also flip and flap. It is very important to see that angle and try to understand. We also have got an elaborative assays in a fly model whether female hormones, but unfortunately Drosophila has got the estrogen receptors. And nobody knows because there is in fly brain, that is what, certain things fly is fly. We need to understand that. But it is important to know when we make a friendship, it's not all that 100 things I accept with my friend, right? Wherever there is a matching, we will go ahead. Similarly, as far as the estrogen story is concerned, Drosophila, I do not know. Estrogen receptor is there, we have been talking about it, but estrogen receptors also involved in the neuroprotection and neurodegeneration. So it is important to look at, but we have a preliminary kind of, a kind of long story with a long age-related males and females data, which is not so, which can be fly-specific. If it is a fly-specific, it doesn't excite us, right? No? But in human, this is the flip side, this is the possibility of the flap. Does it make sense what I'm saying? Yep. Anything else? Yep.
What is the prevalence of neurodegenerative diseases like Parkinson's and Alzheimer's in India, especially in northeastern part of India? Is there any...
So whether it is a south, north, northeast, human is human. Okay. Aging is a systemic process wherever we are, right now, unless we leave the place, I don't know, where the gravity is not there. Okay. So the point is, overall people say that, that's what in the first slide, what is that, after the Alzheimer's, maybe, this is the percentage of the population. Overall we know, but as far as the India is concerned, even if it is the northeast, the same, systematic screening is not done the populations. Most of the time, even, my personal opinion is, though I presented it from a review paper, what is that, India, the prevalence rates are low. So what they will say, perhaps it is because of our food habits. My point is, when I take the exam, then only grading, no. Yes or no? Who has done systemic screening of Indian population, particularly old people, in tens of villages? I was from Andhra Pradesh, I am from Andhra Pradesh, in the villages, visual hallucinations, very common thing among the 60, 70 elderly population. Memory loss, very common. Tremors, even people, they do not know it is Parkinson's disease. People say, he's over, battery is out, so he's, behaving like that. He's old man. So, in the absence of the systemic screening, like it is a wishful thinking. Many times in science that will happen. So there is no systemic study, even the critical diseases like cancer, maybe cancer now people are, started doing it. But for the age-related, late age-related diseases, as far as I know, maybe people must have done in Bangalore, people must have done in Chennai, Delhi, Mumbai, Calcutta. But I don't think in India we have any systemic study. In the absence of that, yes, I put it in the slide. That is maybe from paper I must have picked it up. But my personal opinion is, it is very hard to say. Given, if you allow me to comment, I feel, these diseases like PD and AD, the prevalence in India, it must be much, much higher than the US. The reason is as following. See the quality of the environment in the US. We know it is sporadic, right? In US, if you are there, every 15 years, whether you want it or not, through your door, that fellow will drop that slip where it shows your water during these 15 days. It contains this. Without that, they will never give the bill. Whereas in India, when people, they know that well water drinking habit, rural living, it is also linked with the Parkinson's disease. What does it mean? It has got to do with the quality of the environment. In village, if you go, even in Assam, the guy will come, he will put this much of the insecticide, whatever he will dump into it, he will go. People will happily, they will drink. So therefore, it is very difficult paradigm. In all the developing countries, I will say all the poor countries, the prevalence of the neurological diseases must be higher than the developed world. Developed world, people are very careful with the environment, right? Does it make a little sense? Yep.
It was just a query, because we get to hear a lot of cancer cases, but neurodegeneration as such, I did not get to hear in any of my relatives or friends or anybody. So if which is the
Okay, no problem. I also do not know, but these certain things on logically we can guess, right? Isn't it? Yeah. Okay. So it's fine, no? Okay. So the point is, when the story is coming to, initially during our survival days, we want to do the things quickly as much as possible, because that is also important. Here the grant comes 3 years, sir. So by the time we start, 6 months are over. After second year, we'll have all the problems. So essentially, we'll get the money for the 2 years. So third year, there will be issues. First submission of the UC, it is being regarded. So first we wanted to do the things quickly. But once we little bit relaxedly, you can think. So the question is as following. So entire excitement slowly coming to the mitochondria, and the mitochondria and endoplasmic reticulum also people are linking it. It is not that, so when something is exciting, which is mitochondria, so definitely it is a kind of established. So is it possible the same guy, because we also need to make a thesis at the end of the, it's not that all the crazy things we do that we don't have any story to tell, thesis will not be ready. You may be excited, student will be, he'll be tired and frustration. Yesterday somebody was telling, naturally the stress will come. For me also stress, if a student will come, good morning, if you say, what exactly is going on, please say good morning, even if you are not happy, say that. Okay. So the point is, we thought, okay, why don't we do the same thing? Idea is as following. If mitochondria's involvement in neurodegeneration is known, it is not that something we are saying it, but still, even if it is a known process, can this guy protect? Who is that guy? Once again, the good guy curcumin. So here another scholar, what they did, one important story I want to say, thus for your this one. See, for example, the rotinone, the story related to this part of the presentation. Rotinone, not only in Nagaland, even in Andhra Pradesh, children, it is a plant product. In Nagaland villages, so it is very common. I don't know about Assam and all, I'm sure it is the same, because neighboring areas, induction effect will be there. So you know what I'm saying. So they throw this root into the water. Fish takes it and it paralyzes and it floats. They will catch it. N number of the cases we can see it. But what is that? And most of the time, people believe that anything that is plant originated, it is not toxic. No. The plant, I fail to get that botanical name, but that is a rotinone. That is the root from where they extract the rotinone. Rotinone is a herbicide. What can paralyze the fish? Fish is a vertebrate. Fish is a, it's a very strong animal. Fish, when it can paralyze it, it can paralyze the human brain too. So what is the take-home message? It is as following. People, they'll get it. It's a local fish. Local fish is a very, delicacy. So children, they will catch it and they go around the home and they sell it. But how it is paralyzed? It is paralyzed through rotinone. If you eat that fish chronically, rotinone is mitochondrial complex 1 inhibitor. For the people who are not familiar with this mitochondrial system, mitochondria is a kind of generator, right? This generator, it requires the fuel, you put it and power will come. It's like ATP synthesizer, right? There are two complexes, there are 2-3 gates are there. Whether you close first gate or second gate or third gate, you cannot go. So complex 1, once it is inhibited, your energy production will be down and mitochondria dies. If there is no energy producer, cell will die. In simple, that is the case. So the point is, even what we think, these plant products, it is not neurotoxic. They are very much neurotoxic. There is a very possibility in the villages, people, this neurodegeneration happening because of this fish catching habits and eating those fishes. This is the guy rotinone. So similarly, to make the story short, once again, actually we have got another story also, as I mentioned to you. Like we are also, when the moment we got to know the curcumin, this one, yes, we thought about it, how to handle it. That part also, just I will introduce that part, sir. And we thought, what are the possibilities, even if a guy can, we have certain plant extracts, we did it, following the same protocols, methods, and we optimized it. But this is relating to the rotinone story. As I have described, relating to the neurotoxic and paraquat, everything elaborately assays were performed, and we have picked up in such a levels of the concentrations in order to develop a model where there is no, as much as possible. It is very unlikely. If I take water, it is not that it will go only, one particular place. Same thing with the toxin. But as far as your phenotype is concerned, if it is not affecting, that should be good enough for an animal model. So that is the point. So the rotenone is a mitochondrial complex 1 inhibitor. When complex 1 is inhibited, energy levels are depleted, leading to death of the dopaminergic neurons. Many are sensitive. Dopamine is more sensitive. All important fellows are oversensitive, because this dopamine is such a very important thing. It itself is a pro-oxidant. When it generates the free radicals on its own and combining with the failure of the mitochondrial mechanism, the death of the dopaminergic neurons, it happens quickly. That is one of the possible reasons why there are so many people in the room. How come only 2-3 people are very sensitive? Because there is some inbuilt mechanism. By itself, the dopamine per se, it is a pro-oxidant. So, okay. This curcumin, once again, just to copy and paste the story, just to, in order to give a kind of glimpse what exactly is going on in the lab and to make the story. Here it is health span, it is neuroprotective, dopaminergic neuroprotective, and it once again it is failing during the transition phase where it is supposed to work, which further makes sense. It is very unlikely if a general free radical generator like a neurotoxicant, when it fails, it is very unlikely if someone is specifically mitochondrial complex 1, which is critical for the health of the dopaminergic neuron, guy would protect. Having known that, but still, because we want to tell one more story in a parallel way, we tried and we confirmed it further. This is the limitation. Okay, so again in that piece of work also, we have entire data. I have started writing the thesis where your dopaminergic in all the phases, neurotransmitters, all the phases, everything we have. So end of the day, the driving point is limitation still exists. So simultaneously, once in 2014 onwards, we started thinking, yes, guy is very good, very useful, but he has so many problems. Can we help him? That is the idea. In real life situation also, he is very good, but he has some problems. Can we help him so that we can get help from him also? So we thought curcumin is almost 120 clinical trials already. It is there, isn't it, sir? So it is such a multi-target guy, amazing thing. So can we make him work so that it, to make it as a therapeutic agent, it would be fantastic, because so much is known about the pathways, downstream, upstream regulators, interacting partners. So it is only making the guy being active. He started feeling drowsy. Maybe he's tired, little bit, encourage another only 1 hour. Please don't sleep. Isn't it, bio? So we can say that, and we can encourage the guy, and we can do the same thing with the curcumin. That's what we thought. So this is actually, we tried to put the story 2 times, DBT rejected, 1 time someone rejected. End of the day, it got the Department of Science and Technology. They thought idea is nice, already you have a model, do this. Yes, we started it. But we have what we have done, once again, to make the process quick, we split the story into 2 groups. Adult, make the guy when he is already, adult guy. That means, when he is 50 years, can we make it to work? So by adapting multiple strategies, we tried with a preliminary insights during the late health span phase. This is more relevant, this more excites me, because it is a kind of similar to the human situation. As a component of the food, we adapted a different way to feed to the Drosophila. And as a food component, we tried during the late health, that means maybe when somebody is 20 years, 25 years, and the result shows that it is conferring the protection. We also have a preliminary data about the dopamine and its metabolites. This gives a great hope to us. Yes, he's a Superman. So there is a way this Superman, keep him super by multiple strategies. It depends on your strategy. It can, and it makes more sense for me because No, in Asian civilization, we have been taking it as a food ingredient, it is not as a medicine, right? But if it is a food ingredient, it is not that one day I will take it, I will stop it. In a curry, if you are taking it, people keep on taking it. But if it is when your health span, if you can interfere with the curcumin, if it is protecting the dopaminergic neuron, the late, it makes perfect sense not only for the curcumin, any food ingredient or any nutraceutical. So, but critical question remains, how this changed paradigm is keeping the genetic targets of the curcumin active? Number one. Number two, it is not necessary under this regime, it is acting through the same molecular targets. They can be different. That would be exciting. That would be exciting. If we know when you are old, there is a different genetic target, there are multiple things can be done even to address the problem or address the question of the aging also. But it is important to, it's a long hit, but it is doable by taking advantage of the Drosophila genetics, because we have a switch system. Only when you want to express a particular gene, when you are 50, you can do that. When you are 20, you can do that. Only during the development, we can do that. That would be an amazing, it's a lot of work, but if somebody wants to, it's not that we will address one question, there will be another 10 people, they will address another 20 questions. And we also have been working on, is it possible in the prenatal development, if you can interfere when it is adult, that means if mother is consuming it, is it possible, in the adult stage, neuroprotection, delaying the neurodegeneration at the transition phase, that work also we are doing it, because we are of the opinion, maybe perhaps in the development targets can be different. So it is important to understand critically the question, so that whether it is a curcumin or any other drug, they can be used eventually as markers, whether it will work or not. If it works, whether it will work in the prenatal, or it will work the postnatal, or will it work during the health, or will it work during the transition, or is there any possibility to the senescence when it is 80 years, 90 years, it is possible to develop a marker. Just with the marker, you can screen any compound and you can say, it will work, it will not work, it will work, it will not work. That would be a fantastic thing, but it is a long, this is the idea, sir. Will we do it? We need not to do it, right? Like something we will do it, something someone else will do it. But that is the underlying mechanism. That's why we have taken a lot of effort. I feel it is a nice way to understand it critically. Okay. So there are the other things we are doing it, but the fundamental question is, hundreds of laboratories, we know the free radical theory of the aging or neurodegeneration. So people say that it is a sequestration of the oxidative stress confers the neuroprotection. So for that there is already established markers. 10 markers or 1 dozen markers are there. People keep on screen and they say that this is neuroprotective, that is dopaminergic neuroprotective, this is protective. Yeah, maybe protective, but let us first do the experiment and understand it, and let us conclude the story that way. And here what we did it in simple, whatever the popular markers people have been using it in the animal models, it may be the Drosophila or mice or the C. elegans, all those assays in an elaborative fashion we have conducted during both the phases of the lifespan. To cut short the story, where the curcumin, this is once again we came back to the curcumin and paraquat mediated neurodegeneration. So if the guy is he is working, guy is not working, so what exactly is happening for the established concept which is understood through oxidative stress markers? So this is protein carbonyls. Everybody knows, there is no need to elaborate about it. Carbonylation of the proteins, which is a natural process in the aging process, not only during the aging process, even during the oxidative stress mediated all kinds of diseases, including the Alzheimer's and Parkinson's, which has been shown and which is established. So then peroxidation of the lipids, which is known, we have done. Then the thiols. So thiols in simple, they are good guys, and they help the neuron to be healthy. Then when we perform, the point is like this: the upregulate, the more protein carbonylation, the more peroxidation, and the depletion of the thiols, it happens, which is not good for the dopaminergic neuron, which is rescued during the both the phases, that means health and transition phase. Yeah, the reduced glutathione and the acetylcholine esterase. So these markers, when we do that, the reduced glutathione is reduced under the neurotoxic condition, which is rescued in both the phases, and acetylcholine esterase it is repressed and partially it is rescued in both the cases. And even the popular antioxidant like superoxide dismutase and catalase, the upregulated superoxide dismutase were optimized, and catalyzed upregulated catalases, all these assays are performed exclusively in the brain tissue of the 5-day and 45 or 50-day old fly brains. So the upregulated catalase is optimized, suggesting the established hypothesis is correct. That is how it is working. However, there is another question remains. If the sequestration of the oxidative stress were to be responsible for the dopaminergic neurodegeneration and rescue by the natural product or the food ingredient like a curcumin, how one would explain the sequestration of the stress but failure to protect the dopaminergic neuron? Still, the half of the question is open for the discussion. So, we are of the opinion that sequestration of the oxidative stress may be one of the critical factors which may be essential for the dopaminergic neuroprotection, but per se it may not be sufficient enough to push any molecule or a therapeutic, any drug as a dopaminergic neuroprotectant for PD mediated pathology. This is the stage where we are standing today, and it is, the story is ready. That is what we are, how it would go, we need to see further. So if that is the case, what is the next step? Next step is try to understand the molecular targets. What we did it, maybe we picked up around 38 to 40 molecular targets, because somewhere we need to start the story, and we try to look into at the level of the RNA using the quantitative real-time pace, the transcription levels. But actually we messed up. Many of the people, they have shown that JNK mediated, even at the level of the transcript, at least that one pathway is it's a canonical JNK mediated neuroprotection. Even at the level of the transcript, it is upregulated, but we don't see that even the JNK downstream, the FOXO and the another HSP like gene expression profiles, at least at the level of the RNA, we don't see any difference. Still, it makes sense for me, because JNK, though people have shown whatever is published, it need not be correct. JNK and JNK phosphorylation are critical processes for the neuroprotection. Therefore, before we conclude it, the protein and phosphorylated and non-phosphorylated, even if it were to be the similar, it is also important the per ratio between the phosphorylated and non-phosphorylated. Then only conclusively we can say that it is not through that pathway. Okay, this is the FOXO, and that is the downstream once again EFL. Okay, then maybe we have taken FOX transcription factors, oxidative stress response, iron homeostasis. Overall, they fit into the 5-6 pathways, and we have a kind of the transcript, the RNA level expression. But when we see entire data, it is yet to be, conclusions have to be made before we substantially make certain decisions. I am of the opinion that it is not exciting. It is not that something we are seeing different. Maybe the curcumin may not be, it is working, it is not working. I don't see any intelligent clues there. So I thought it is important to see all the 30,000 transcript in the brain specific transcriptome. We went ahead and we have taken advantage of the Illumina platform, and we have got it done under all the 12 groups belonging to the 2 subgroups relating to the ACE. And the story is, yes, still this also further confuses us, but that is a natural process. When we, I'm sure, sir, you know that when the Affymetrix chips were there, validation has been the critical issue. So it has to be validated, whatever we are seeing it. Then it will be definitely, there will be excitement, which is very unlikely, all this effort. And yes, we also see certain, may not be the transcripts, they vary it, but isoforms, there is every possibility the differential splicing mediated neuroprotection is very much possible, which has been known even in the human brain. So there are certain, when we see the rescue and the diseased brain, we see the significant level of the novel isoform variation. Here lies the great opportunity. If we were to figure it out, even 0.1% variation, that would be interesting story to the global story of the dopaminergic neuroprotective strategies. Okay, overall it looks very simple. Actually it confuses further because this fits into 20-25 pathways anywhere and everywhere. But this has been the story, even Seymour Benger's 2002 paper, where they have shown the age-mediated transcript regulation. It fits into all the pathways. Age is a systemic thing. All the differences, all the multiple pathways we see with reference to the age not necessarily need to dopaminergic neuroprotective or neurodegenerative. So it is very important intelligently, we need to subtract and pick it up. It's a long hit and validation with the taking advantage of the Drosophila genetics. If not we, someone else can do it. This is the stage we are, but certain pathways, it makes sense for me, like a microtubule, or we are thinking about it, we need to work on it and make sense out of it. Before we make sense out of it, we need to validate it thoroughly. This is the stage as far as the transcriptome data is there. We are, but certain things, yes, it gives a hope, right? When the time goes tough, even a little bit, candle light can be better than suns. So here we see certain, in the literature when we dig it, the brain-specific nucleotide phosphatase kinase activity, which has been shown differential expression, which is already there in literature in case of the Alzheimer's disease, and even enhancing nucleotide metabolism protects against the mitochondrial dysfunction and neurodegeneration in a PINK1 model of the Parkinson's disease. PINK is a kinase. It is also involved in the Parkinson's disease pathology, one of the genes that is described. So the targets also fit into our data, suggesting it is very unlikely the neuroprotective or neurodegenerative pathways, only some pathways will match in your data. It is very unlikely all other things may not provide any clues, but it is important to understand it, digest it and make sense out of it. So then we try to understand transcriptome, global transcriptome. We are working on it. I should not say we are trying to understand. So then we thought it is important. This is the time we have the methods established. All the things we are a little bit comfortable. Can we do the screen? This is what exactly, for me personally, it is an exciting story because the power of Drosophila is its genetics. What we did to begin with, we thought we can do the, all the, as I mentioned yesterday, Drosophila has got the 4 pairs of the chromosomes, Drosophila melanogaster. So we started looking into, a chunk of genes that are gone, so by suppressor enhancer screen. What is suppressor enhancer screen? In simple, either the phenotype can be suppressed, that means there is a good guys are lost, or it can be enhanced, that means either the dopaminergic neurodegeneration can be worsened or the protection can be improved, suggesting the possible players, whether it is that there may be good guys, there may be good guys are lost, or the guys who are not good, they are gone. So idea of the suppressor enhancer screen, which has been established, it's a lot of work. Still we are slowly moving in that direction. And yes, some lines it gives a clue that hard effort is not a waste. And we also see which is known player of the dopaminergic neuroprotection or the neuronal protection, like the centrosamine. That specific line which validates our screening strategy is not a wrong thing. There is a possibility provided carefully, because it's a lot of effort and understanding about the experiments, making the crosses, bringing it out, scoring the specific genotype, and understanding it further. It's a lot of effort, particularly for the graduate students, but still we are making an effort. in that direction. Yes, this is another one. Yeah. Okay, simultaneously, again, as I keep saying, stories also should come out for the scholars. They also, yes, project and thesis is a different thing, but it is also important. We started looking into is there any whole, we looked into the whole brain specific proteome. Then phosphoprotein levels, the variation in the phosphorylation processes under the disease and regular, which is very much known. So this is the global protein phosphorylation levels. Certain things we see it, but it is also important wherever we see the variation, because it is a global, we need to figure it out. If it is a 50 kilodalton, any one of these guys, maybe it is a tyrosine hydroxylase or any of them of the known players, they fit into that particular weight proteins, and it is possible to understand it further. And tyrosine phosphorylation is a critical amino acid phosphorylation, which has been implicated in dopaminergic neurodegeneration. So we also once again we took it, but what is important is, it is very, very important to at least some probable players of the neuroprotection or degeneration, gene to gene, perform it and validate it. Yes, glycoprotein levels, once again, protein calculation is a critical process implicated in the neurodegeneration and the neuroprotection. So therefore, once again, it is performed in both the age group flies under the rescue and non-rescue conditions. It is also important, maybe other day Dr. Patel, he has shown at the level of the putative transcripts, so it is possible rather than running the gels, if you can do it, that's what actually we are thinking now. At least certain candidate things we can, this is the stage we are about as far as the glycoprotein levels are concerned. So with this, our neurodopaminergic, neurodegenerate, brain-related story, it is a kind of, I said we have got other stories, but it's a parallel stories. This is what the stage where we are. If there is any specific question, I'll take it. Otherwise, I would move on quickly. Anything, particularly people who are not from our.
Have you or can you compare curcumin with whole turmeric in your system?
We have not used the turmeric. We have all these experiments are curcumin only.
Can you?
Definitely we can do that. Definitely. And the only thing is, if this is my gut feeling, if it is a turmeric, it wouldn't work this way. It has to be within the food. It has to be.
Because there are over 200 different bioactives.
Correct, sir.
Turmeric. So the question is, will the whole turmeric be as good as?
Yeah. That is definitely manageably doable, because already we have come thus far. It is manageably doable. Yeah. Okay. So most of the times, that is human behavior. Instinctively, or maybe by learning behavior wise, what is very, very important for us, it is the same, I think, any animal, even the cell, at the level of the cellular level, right? What is very important, we always try to focus, because cognition is very important. So most of the time it is the brain related story, in whether it is a Parkinson's or Alzheimer's, people have been trying to understand. But what is known, there are multiple phenotypes which are not linked to the brain, are also present in case of the Parkinson's, which is like, for example, loss of the constipation, digestive problems, and even sexual dysfunction. Sexual dysfunction has been known in PD patients. So, but what people they do not know is, because there is not lot of work is done on sexual dysfunction in PD. The beauty with the Drosophila is essentially my training was the behavior genetics. So Drosophila is a fantastic system. One can characterize the courtship traits and sexual reproductive traits in a precise manner. The genetic mechanism, that is underlying genetic mechanism for all these phenotypes, the genes, they are conserved between the Drosophila, mice and human. So therefore, that time we did not have much money. Somebody said another candidate. So we thought how we can tell the story, without spending much. So that is the idea. In a small way, we started looking into the sexual dysfunction. So sexual behavior has been eternally, it has excited all the animals, more so humans. So we are excited about the sexual behavior, all the prophylaxis, how we can enhance the sexual capability, under the disease condition, is there any way to the rescue phenotype? Eternally and instinctively that bothers us and excites us, because like memory and cognition, reproduction is one of the critical things that is, it is for the survival, because reproduction and survival, reproduction, mate selection, sexual behavior, they are interlinked characteristics. So for this, fly can be of the great help. So we started in a small way. Drosophila sexual behavior has been characterized, courtship behavior has been characterized, and mutants were screened, certain genes were deciphered in fly model. So it is like when two flies approach, this is very self-explanatory. This is from a nature review, when a fly, two flies approach, and it is very important. It is not that Drosophila, it will mate with anybody and everybody. It is not that Drosophila female will allow any male and every male, it will allow it to be mated. There is a sexual preference in Drosophila. It is not that sexual preference is there in Drosophila, sexual preference is there all along the evolution. I am sure among if there are two different strains, in the E. coli also there must be sexual preference, there must be a mechanism which we do not know. So the Drosophila, the male, the one with the dark, it is the melanogaster. Yesterday we were talking about it. So this approaches and it makes all kinds of, courtship behavior in order to impress. Essentially, it is the male which plays in, I think, I do not know, some birds and all, there are some exceptions, but otherwise it is all the time, it is the male is the active partner, and it will try to impress. Sometimes a female, if he likes it, okay, if he doesn't like it, still this guy, he doesn't give up. So, and in the end it leads to the successful mating. That is the long story short. In the human also it is the same. The courtship traits and certain genes, there is a conserved genes between the Drosophila and the human which are involved in the courtship. But what is important is, it is not that only the courtship, it is only it is in the courtship. There are other phenotypes, critical phenotypes also regulated, including the development, but same is the story with reference to all the neuroprotective genes, all the genes that are involved in cancer. For example, NF-kappa B. NF-kappa B is critical for the very development process itself. So, what we did when Javilo started in a small way, what the point is, okay, what is the idea? What we thought is it possible before the guy trembles?
Sexual behavior, when you talk about sexual behavior, is it male or female? Because there are different hormones that like more than one versus the other.
Correct. So, whatever the data I am presenting today, that is with reference to the male sexual behavior. Male sexual behavior. Because yes, we also need to do, maybe slowly we will do the female also. Number one, it is Parkinson's is more prevalent in male, so it is good to start with the male. And it is relatively easy to characterize the phenotype in development male. Female, there are certain factors are there. So to make the story simple and handy for the students, we started with the male. Okay, is it okay, sir? Yeah. So the point is, what we thought, yes, there is a resting tremor, mobility defects. we are doing with the sexual behavior. It is not that just we want to, it is another phenotype we want to do that. Can, is it possible to screen the PD phenotypes before the onset of the mobility defects? That would be a fantastic thing, because by the time you see the mobility defects, 70% of the dopaminergic neurons are degenerated. Neuron being a post-mitotic cell, when 70% is gone, there is no way you can develop any therapy. That is the critical limitation to develop a successful therapeutic strategy for the Parkinson's disease. I am sure same thing with any other neurological disorders. So is it possible? Maybe this phenotype is, it is not possible. Can we see the another phenotype? The phenotype what we are seeing is the sexual behavior. So we thought, basing on the courtship, is it possible? Because genetic basis is known, is it possible to screen those genes even in human population? Is it possible? So that is the idea. I do not know how far we would go, but with that idea, we have a fly PD model, which I explained it before, and we have developed another specific fly PD model just to characterize the sexual dysfunction phenotype. What is the idea? The take-home message is as following. Can we characterize the courtship traits and the regular and abnormal under the induced PD condition when the guy doesn't tremor? That is the idea. So we did an array of the experiments and we picked up such a window where guy doesn't tremor. Okay, that is what we want. We want him to be, tip-top. But still, if you can if you can pick up a sexual behavior, the courtship marker, yep, that means this is a kind, it need not be a biochemical marker, at least at the animal level it is a behavioral marker. There are multiple behavioral markers, including the negative genotoxicity assay. So we characterized that, and yes, the guy is not trembling. We took advantage of him, and the multiple copulation duration. For the people who do not know, it is a very self-explanatory, and percentage of copulation, how many guys out of given number, and courtship latency, the time taken. You have seen the original picture, courtship latency, even the male and female, when you put into that mating chamber, it takes some time. Then first when it taps, that is the latency period. Oh, okay, guy is started. Then mating latency, the time taken to mate, and attempted copulation, how many times, NSE, non-sexual behaviors, tapping, scissoring, vibration, and circling. There are innumerable number of the markers are there. Out of that maybe critical 8, 9, 10, we worked on it, and when we characterize it under the induced PD condition, when the driving point is as following. When there is no dopaminergic neurodegeneration mediated mobility defects are not observed in Drosophila, courtship markers can be characterized and variation can be understood in the same model system, which means this can be of some help, in order to, is it possible to take it to the next level? We do not know. As of now, Javilo started writing thesis. We'll see how it would go. As you said, even female also I want to understand, there should be a match between these two, then only we can. And yeah, and importantly, we also, the data I have not included it here, we also with the curcumin mediated sexual dysfunction rescue. Yes, curcumin, by following a specific regime, curcumin is rescuing the courtship behavior trait markers under the induced PD, and we also know it is perhaps it is dopamine mediated, which makes sense because dopamine's involvement in the sexual behavior is known. Degeneration of the dopaminergic neuron leads to male and female sexual problems, which is also known. So, however, again, I do understand there is a limitation because all these experiments are done during the health phase. Will it protect? There is every possibility because it depends where the hit at what stage. If it is if the dopamine level is marginal, perhaps curcumin may work. It may not rescue the mobility defects, but there exists every possibility. It may protect the sexual dysfunction. Still, it will perfectly make the sense. So this is what the stage where we are. So now we know, like, many kinds of foods and yoga, meditation and exercise, listening to the music, trying new things out. It's very important. Everything gives a dopamine shot, dopamine. That's how, now figuring out, as Freeman said from Caltech, the great Nobel laureate, figuring out the new things is very important. That's what Nancy Bonini writes, Seymour Benger's obituary. Seymour Benger has got a peculiar habit, that went into his science also. He always wanted to try the new food. He always wanted to go to the new restaurants. So it is not only that, whenever some graduate student or a postdoc comes, he always says, it is known, what is new about it? That is maybe perhaps that intrinsic, that I look into the regions where nobody, bothered or dared to look at it. Maybe that is very important to do the, novel or virgin science. It is important, untouched areas. I think that is why Seymour Benger, maybe it is a physics or genetics or virus or Drosophila or sexual dysfunction or a pain or neurodegeneration. There is no area the guy did not touch. So I think it is important to figure out that habit is a nice thing, for which dopamine is chocolates and coffee and citrus fruits. Of course, tomatoes, my favorite, cheaper, you can get it. Why it is citrus fruits and all, they may be expensive. Yeah, green tea, once again, typical Asian. So, yep, it is important to figure out. Okay, so it is not necessary to once again to recapitulate whatever I said it. I hope if you have some questions, I will take it. Otherwise, I profusely thank funding agencies, particularly the DBT, UGC, ICMR, DST, Nagaland University. Heartfelt regards to Stefan, Jose, Joan, Min, Fiona, Fishbeck, my supervisor, doctoral advisor, Professor Hegde, and Professor Vijay was when we were hard times, he was our advisor. That time he was in NCBS. In 10 years in 10 years, from professor to DBT secretary to the advisor to the government of India has become. Of course, I also became professor. So lecturer to reader to professor. Then our collaborators, Dr. Muralidhara. Actually, initially we wanted to do the Smita's lab, the brain imaging. That was the idea, but we could never do that. And for the screening, Seshi is our advisor. Grish gets the lines from America to there, and we'll go and bring the lines back to Nagaland, because here there is no way we can, in spite of that 50% of the lines are every time lost, which is a very common thing. Yep. So with this, I think it's an overview of our, if there is any questions, we'll take it.
No problem. We are possibility in the field experience to try this thing? I want to know.
Actually, it is already being done. Already it is being as a therapeutic agent. As Sir has been elaborately has been telling in all kinds of cancers, it is in every cancer it is the inhibitor of the NF-kappa B, for example. Even in the neurological diseases, not only there are 120 clinical trials it is there, it is already being used as a drug. Curcumin. Yeah.
My point is, yes, in the form of drug, yeah, the doctors are referring that many of the drugs provided license, that is one of the things that we are commonly experiencing. But as a scientist who are well versed in the subject, I think in one sense, what doctors are providing the medicine, the main details inside what is going on, all the symptoms is given by, I think, the scientists.
Yeah, Sarat. Yeah.
So in that case, excluding the doctors and the prescription, is there a possibility we as a scientist try to provide our patients that or this Drosophila?
Yeah, in simple, it's a long question, madam. The point is, rather than as a therapeutic agent, as a drug, maybe it is important to push. Of course, our very lifestyle is inbuilt in such a way as a prophylactic, which makes perfect sense because it's a food ingredient. Even not as a, it is not. Yes, it can be a drug, but rather than as a being used as a drug, maybe as a prophylactic, as a food ingredient. It itself is a food ingredient. I think it is important to popularize and make the people to, that would be a great help, because though many people they know turmeric is good, I don't know how many people really taking advantage, including in India. So maybe that may be of the more good. Even our results also show that it can be a fantastic prophylactic agent. It will do more good as a prophylactic agent. So therefore, I think that angle is important. This is my opinion.
So there are problems. Suppose sun has produced all the detailed information. Okay. Later on, drug will be manufactured by somebody, and that particular doctor will give the prescription.
Yeah.
Drug. And then now pain will come because of this treatment that patient is cured or something like that. But the mastermind behind is research knowledge. So in that case, this I may be wrong, but I just will observe or yourself also like you can provide an advice to our common maybe surrounding and provide this thing, and if we could make out the improvement and then we popularize. Is there anything restriction or this thing that I want to clear?
Actually, Sir has, I listen to many people who are working on curcumin. Maybe very few people, maybe 1% of the people, Sir has been pushing one point very strong actually, even in this course time or otherwise even privately also. Rather than a curcumin, and he says he analogy, if it is the turmeric is $1 per kilo, curcumin maybe, maybe I don't know, 10 times or 20 times, drug maybe 200 times. So what he has been, and what is important is pushing the turmeric itself is good enough. That is what, that will take care of it. And even if Sir wants to refine that, if you want to comment on it, Sir, I think you should. So it is not the curcumin, turmeric per se, and people have shown without curcumin, Sir also has presented the data, even other components together, it has the same effect. Even if you take out this curcumin, there are other 50 fellows, his friends, and there also can be neuroprotective or anti-carcinogenic. So that is the point. So best thing is turmeric also can be, like the way, rather than the hype being on the curcumin, I think real hype should be on turmeric, which is very, very affordable for every one of us. Yeah, I think, sir, if you want to make in the...
Yeah, no, I just want to comment that doctors never fail give the human to the patients. And the reason for that is neither the doctors nor the pharmaceutical companies stand to make any money on it. FDA has already approved curcumin. If anybody want to use it, they have no concern. They declared it as a grass, generally regarded as safe. I have hundreds and hundreds of patients on curcumin, hundreds of them. And they all contact me, and I have an SOP, meaning standard operating procedure, and I just pass it. So there are 350 clinical trials to prove that it is working. Now it is left to the patient, and patients are not blind because patients need help. So therefore, they read everything these days online. So they can very quickly come up with what we know and what we don't. So they start, and safety is not an issue, cost is not an issue. So therefore they start taking it their own. And then they write me that what it did for them. So therefore I have a whole database based upon what patients tells me what the human did for them. And as I mentioned that till today, to my knowledge, no cancer is known that is not affected by it. At least in the test tube, at least in the animals, and maybe even in the humans. And in humans, unless you try it, you will not.
Yeah. Yep.
You had Dr. Bharat gave us an incident in which somebody made curcumin fluorescent, and we found that it reached brain the number 1.
Curcumin per se fluorescent. It is not making the, isn't it, sir? Curcumin per se fluorescent, one of the chemical.
I mean, whatever. Something happened that he said it reached the brain as at the number 1 spot, fastest. So, connecting that statement to dopamine, do you think there is a chance of something more to do with dopamine with the help of curcumin because of that?
No. I think we are mixing up. Sir's point is with reference to the bioavailability and the permeability to the blood-brain barrier. What popularly people, the argument against the turmeric or the curcumin is, bioavailability. People always, nobody really worked, properly in the kinetics and everything. Simply they said, curcumin, he's a good guy, but, he's crazy, something like that. It is very easy to, write off any person. Same thing, people say curcumin is good, but it doesn't go to the brain, it cannot pass through the blood-brain barrier, and bioavailability is the issue. So with reference to, Sir has been making a strong argument. There was a study in Harvard, that's what you said, right, sir, and they have scanned the brain even within 15 minutes or 20 minutes after the consumption, it is shown up the signal in the brain, but it is very unlikely, I don't think Sir said it is exclusive. No, curcumin it goes to everywhere, but the question is with reference to crossing the blood-brain barrier, which is happening. That is the argument. So, with reference to the dopamine, yes, if somebody is, when you are addressing a neurodegenerative issue, crossing the blood-brain barrier is important because where it is supposed to work, it has to go there, right? If it doesn't go, you are a good guy, just you are sitting there, there is no use. Where you are supposed to go, there you have to go and fight. So once the curcumin enters through the blood-brain barrier and it goes to the brain, that's all it is. So guy is there where he is supposed to be, that is the idea. So there is no problem, that is the point. So that's why the curcumin analogs, whole concept of this, I don't know how many million or $billion industry, curcumin analogs have been so popular, just they will change it and patent it and they say this can be a drug. But there are also other issues. What about the biological efficacy? Yes, people say it, we modified it, it is biologically equally efficient, it is not toxic, but when curcumin per se can do that, why you want to go for the analogs or all those things? Maybe that is all the time it is the drug industry, right? One is good, two is good, you cannot patent it, mix it 1 + 2, you can patent it, right? That's where the money is.
I was trying to drive you to develop a drug which he wants us to develop. That's what I said. I was giving you the logic.
I think this you should discuss with the sir about developing a drug. I have no idea.
Because you are working on dopamine. That's what I am telling you.
Right. Point is, yes, curcumin, Drosophila doesn't have the blood-brain barrier, but now people say it has got a similar structure, but may not be as strong as it. But when studies show that it can cross through the human blood-brain barrier, whole discussion should end there. That's what I feel, that this is my opinion. Yeah, because that is, if it, see, the point is where it is supposed to work, if it is not reaching, I think whole thing is gone there, right?
What after reaching? It has reached, that's what I said.
That's what now we know that it is working, right? Once it reaches, we know that it is working. Whether it is a cancer or a neurodegeneration, we know that, right? And even traditionally also we know that, and particularly, I don't know here, even in my hometown in Andhra Pradesh, all the time I used to take curcumin with the milk. I never took the curcumin in water. We know that bioavailability now, scientifically people, they know that, you know, curcumin always must be taken with the milk. Milk is being, you know, it has the fat component. I'm not a chemist, but it's a common sense. Curcumin, it dissolves in DMSO. But you know, in the milk, when you mix it, it partially soluble. That is the whole idea. That means it is very unlikely when we have so many 200,000 liquids are there. People are specifically, they are prescribing milk, curcumin with the milk in Ayurveda. That means, yes, we may not be having how they validated it, but we know, you know, how it should be taken. I think that's an important point is, you know, yes, yesterday also somebody was telling curcumin if you take it. No, I don't think anybody takes curcumin like that, but as far as at least I have seen, people always take curcumin with the milk, right? Yeah. I think if there are no other, already we are, 12:30, 1 o'clock again. Is it okay? Okay, thank you, it's nice.